All right, I am pleased to introduce today's speaker, Ella Segal. Ella grew up in Bethesda, Maryland, outside of DC, and graduated from Rice University in Houston, Texas in 2021 with a bachelor's in ecology and evolutionary biology. She gained considerable research experience as an undergraduate in Tom Miller's lab, studying grass endophyte mutualisms and conducting meta-analyses. She also completed an REU in the soils lab at the Morton Arboretum outside of Chicago. She began her internship with the plant ecology program in June of last year, and this internship was funded with generous support from the Von Jordan Foundation. Since arriving, she's been involved with data collection for most of our long-term demographic projects and been a part of several conservation activities. For her intern project, she studied seed banks of some of the endemic herbs, and today we'll finally get to hear the results from her highly visible project on the veranda that so many of you have walked by so many times over the last 7 months or so. This spring, Ella will intern at the Smithsonian Environmental Research Center in Maryland, and this fall, she'll begin her PhD at a yet-to-be-decided university. With that, I'll turn things over to you, Ella.
Thanks, Erin. Excited to be here today. So we have these 2 slides. Yes, so as Erin said, my name is Ella Segal, and I am an intern in the plant ecology program. My internship was funded by the Von Jordan Foundation. And my intern project is to characterize the structure of seed banks of endemic species in scrub and roadside habitats. So I came up for the idea with this project just driving along the fire lanes at Archbold. Archbold is home to large numbers of endemic and endangered plant species with extremely restricted ranges. When you're driving along the fire lanes, it's hard to miss how large some of the populations of these species are. I was curious why, with so much available interior habitat, some of the densest populations of these species were on some of Archbold's most disturbed lands. A common link between a lot of these species is that they have a soil seed bank, and because the maintenance and creation of roadways could conceivably disturb the soil, I decided to look into the seed bank further. So first of all, we'll start off with what is a seed bank. The soil seed bank refers to the seeds and fruits such as achenes or caryopses on or in the soil, according to this 1989 book. Pretty much when plants produce seeds, they don't germinate the moment they hit the soil. Instead, they stay in the soil for some amount of time before they either germinate or degrade. If they stay in the soil for less than a year, it's what's known as a transient seed bank. And if they stay in the soil for more than a year or up to millennia for some species, it's called a persistent seed bank. These persistent seed banks play particularly large roles in population and community stability, as they harbor large amounts of species richness and genetic diversity and replenish the above ground vegetation population over large periods of time. The reason I looked into vertical structure of seed banks is that a lot of research has found that that's what's important for shaping above ground vegetation populations. One main reason it's important is because burial depth affects seeds' germination cues. Seeds buried at different depths may experience differing levels of light, oxygen, moisture, or temperatures, which can all serve as germination cues for a seed to either germinate and become a plant or not germinate and stay a seed in the soil. These different germination cues at different depths lead to what some researchers have described as an active part of the seed bank and a storage part of the seed bank. In the active part of the seed bank, seeds are close enough to the soil surface to be able to germinate, or they may become food for animals, get buried to the storage part of the seed bank, or decay. In the storage seed bank, there's really only 2 options for a seed. They can resurface and become part of the active seed bank and maybe have a shot at germinating, or they can also decay. One more thing to consider when we think about Archbold's seed bank structure is that some research of climates with wet and dry seasons have found a bimodal distribution of seed bank structure. And that's because there's a high concentration when seeds fall here, but when in the dry season, cracks open up and some fall through to lower layers of the soil. So putting this all into the perspective of Archbold seed banks, there are 2 things that would be helpful to understand. First of all, which plants have a seed bank in the scrub, and the second thing is a dominant vegetation pattern in the scrub. So these are some thick shrubs that surround an opening area, and this pattern repeats a lot in the scrub. Your shrubs tend to be your oaks, your rosemarys, your palmettos, among other ones, and they are your resprouters and cloners, and some of them combine seeding with those methods. You can see how dense this environment is, and in dense environments like this, there's a lot of competition, which puts individual plants under lots of stress, and resprouting and cloning yourself are less costly methods of reproduction in these environments. So these plants don't have to expend as much energy in reproduction, and that helps them grow even denser. These smaller plants, as you can see here in the middle of the gap, these are your obligate seeders. They cannot resprout or clone themselves, and they depend 100% on the seed bank to reproduce. As I mentioned, sexual reproduction or producing seeds is pretty costly, so most of the time these are outcompeted by shrubs as they grow, and the seed bank stays dormant. So they're outcompeted and their populations decrease. The seed bank remains dormant until favorable germination conditions occur. And in the scrub, these favorable germination conditions tend to occur with disturbance. Historically, this has been fire, as fire either kills or greatly reduces the size of the shrubs, which gives seedlings that germinate from the seed bank the right amount of light and space and lack of competition for water and nutrients they need to take off in population. Prior research from the plant lab have also found that scrub seeds germinate at higher rates in the presence of heat and smoke. And all of these things contribute to larger populations of obligate seeders just after fire, whereas all other types of plants tend to decrease just after a fire. This trend reverses with time since fire as shrubs recover and grow, and again outcompete obligate seeders, which decrease. Nowadays, though, fire isn't the only disturbance we see in the scrub. On the right here, you can see this contraption, which is known as a disker, and as of last week at least was parked outside of the maintenance sheds. It's what's used to create and maintain roadways at Archbold. So these diskers go through on the sandy fire lanes, and they make sure that none of the big shrubs can grow and obstruct roadways. In a lot of ways, this probably operates similarly to fire in that they keep the shrubs at bay, and then obligate seeders have the space to grow, and is likely why it explains a lot of the density of obligate cedars on roadsides. What isn't clear, though, is how or whether these disturbances affect the soil seed bank differently, and whether any differences in the soil seed bank are then reflected in above ground population. So just going back to how obligate cedars do really well after fire, you can see that this area has been very recently burned, and Paronychia chartaceae, this fluffy green looking plant, is very happy for all the space. So in my study, I wanted to get at 4 questions. First of all, does seed abundance vary between roadside and interior habitats? So putting above ground vegetation density aside for just a second, maybe seeds degrade more quickly in one habitat than the other, or maybe individual plants produce more or fewer seeds in one habitat than the other. 2, does vertical structure of the seed bank vary between roadside and interior scrub habitats? So we saw the disker, you can imagine trucks going by, they're turning over a lot of soil. So maybe that's creating a looser soil structure where seeds can fall through into deeper depths more often in roadsides than not. Or maybe it doesn't have that much impact for the roadsides, whatever's happening on the roads. So 3, are variations in the seed bank reflected in above ground vegetation density? Before I started this study, I could kind of see it going both ways, where you might have a denser above ground population that would produce more seeds and then make a denser seed bank, and then maybe even the denser seed bank would lead to a denser above ground population, and that cycle could continue. Or I could see there being just higher germination rates of fewer seeds that perpetuated an above ground population that's pretty dense, but there wasn't necessarily more seeds. And 4, does abundance of seeds correlate with viability of seeds? So just because you see a bunch of seeds in the soil, does that mean that there's a greater chance or greater ability of that seed bank to produce above ground vegetation? So to look at this, I looked into the seed banks of 2 plants. You can see Paronychia chartaceae here on the left, which I've mentioned before, and this one on the right is Hypericum cumulicola. They are both federally endangered and endemic to highlands and Polk counties, and they are both obligate cedars. When I wanted to compare roadside and interior seed banks, as you might imagine, I went to roadside and interior study sites. So on the left, you can see two pictures of some roadside study sites. This roadway had a lot of Hypericum and Paronychia, great site. Here you can see a Paronychia population looking pretty happy, even with some tire treads through it. In interior sites, you can see some background palmettos here. These were your gaps. One way you could quickly tell where some soil came from most of the time without knowing where it came from was the color of the soil, because interior soil tended to be darker with all the fires that had gone through it. And overall, I went to 4 roadside and 4 interior sites for each species, except for Hypericum, I went to 5 interior sites. So this left me with 17 sites, and at each site, I took 4 replicates, which gave me 68 replicates. Each replicate consisted of 12 soil cores taken around target species. I then separated each core into 4 depth layers. So there was a 0 to 2 centimeter below the surface soil layer, 2 to 4, 4 to 6, and 6 to 8. So for a total of 816 soil cores. And then combining these all together, well, not all of them, some of them together, I got 272 Petri dishes and 272 subsampled from those Petri dishes for germination and microscopy, respectively. A lot of you, as Erin mentioned, probably saw my Petri dishes outside the plant lab, some good times watering them. I watered them every day for a little over 3 months, and then I took censuses twice a week to see if there were any new seedlings that had come up. After I identified the seedling to the species, I took that as an indication of a target species seed present in the seed bank. And then for microscopy, I took subsamples, as I mentioned, from this. Each Petri dish had about 100 grams of soil, and each subsample had about 7 grams of soil. And using this plate and tweezer method, I looked through it for possible suspects for seeds, and then I looked at that collection under a microscope to confirm whether they were actually seeds. So here's a Hypericum seed, here's a Paronychia seed. The differences between these two methods in terms of what data I got was that germination gave me the number of viable seeds, because that's just the definition of a viable seed, is can it produce a seedling, which happened here. And then microscopy was just sheer number of seeds in the soil. So results. First of all, seed abundance was much greater in interior scrub than in roadsides. You can see this is true for both species, with a sharp drop off as you move from B, which was balds, to roadside. So there you could see that Paronychia just had a denser seed bank overall, which was true for all of the graphs I will show you. And then putting aside habitat for a second, so lumping interior and roadside habitats together, you can see that in the top 4 layers had a lot more seeds than the bottom 4 layers. So there's a sharp drop off here, similar drop off here. And now putting both of those factors together, you can see that the distribution of seeds did not really differ from roadside and balds, with the top 4 layers having the most seeds and then sharp drop-offs, and this pattern repeated itself a little less here. My analysis shows for Paronychia, habitat was marginally significant, depth was significant. For Hypericum, both habitat and depth were significant. And then if you look at the habitat depth interaction, which is very not significant for both, that shows that there's not a significant difference between the seed bank structure of both habitats. So then after I did that, I was curious about what would happen if I extended my model to include the bottom 16 centimeters of soil, because I had only looked at the top 16. I'd only looked at the top 8 centimeters of soil. So to do this, I calculated the percentage of seeds that was found in each centimeter of soil. And then this is a cumulative curve that shows how many seeds you would expect to find, with 100% coming at the bottom layer, or getting to 100% by the bottom layer. And the curves were very similar for both species. I had originally separated them by habitat, but those were so similar that I lumped both interior and roadside ones together. So the big takeaway from this graph is that by 6 centimeters, pretty much 80% of the seeds had already occurred. And this is probably a more conservative estimate than my study design yielded, because I'm assuming that seeds actually do occur in these bottom layers, even if infrequently, which they might not. But overall, from both of these things, we can conclude that most of the seeds probably occur within the top 4 to 6 centimeters of the soil. So then I also quantified vegetation density when I was at each of these sites. I took a 1 by 2 meter transect, and within each of those transects, I just counted the number of stems of each species. So probably be like 1, 2, like, it's hard to tell with a picture because you might actually have to get in there and feel around for what is an individual plant. But the point is, it was stems per meter squared was my vegetation density metric. And then I related these to my seed counts. So viable seed count did not correlate with above ground vegetation density, except in Paronychia, it was slightly significant and negative, but that was probably driven by this outlier, so I didn't worry about it too much. And then Hypericum was not correlated. For total number of seeds and above ground vegetation density, it again wasn't correlated. And then finally, I turned to my final question, which was relating viable seeds and total seed counts. For Paronychia, this was slightly correlated, but it was a pretty weak correlation, but still significant. For Hypericum, it was not correlated. You might notice, though, that the maximum number of seeds I found using my microscopy method was 8 for Paronychia and only 2 for Hypericum. So it's possible the 7 to 8 grams of soil I looked at under the microscope was sufficient to capture Paronychia's total seed density, but not Hypericum's. So to recap my conclusions, first of all, the upper 4 to 6 centimeters of soil has a majority of viable seeds in both habitats. The depth distribution of seeds does not significantly vary between habitats. Incidence and abundance of seeds is much higher in interior habitat than roadside habitat. Seed abundance and seed bank structure do not correlate with above ground vegetation density, and there was no or a weak correlation of microscopy and germination data. So implications, first of all, the upper 4 to 6 centimeters of soil may be the scrub's active seed bank. We can't really conclude this until we conduct an experiment to see the maximum depth at which a seed in the scrub can germinate from is. 2, with fewer seeds overall, roadside populations may be less stable. So the high above ground vegetation density would suggest that they're pretty stable, but they would have less of a safeguard if something were to happen to the populations. It also might show that the germination rate of these seeds is higher just because it takes fewer seeds to maintain these populations, and maybe more constant disturbance plays into that. 3, total numbers of seeds may be more similar between depth layers, but seed viability might be higher in upper layers. So my data showed that more seeds germinated in the top 4 centimeters of soil, but the viability and total number of seed data wasn't super correlated, suggests that maybe lower layers would have more total number of seeds, but they would just be less viable. My microscopy data also showed that. I just didn't show it in this presentation. And the fourth thing that we were curious about is, where is all the seeds? Because I did not find as many seeds as we thought I was going to find. For my germination data, I had exactly 100 seeds that germinated. And for my microscopy data, which was 93% less soil that I looked at than germination, it was about, it was 101 seeds that we found. So that is a decent amount of seeds. It's good to analyze, but if you look at a plant, like this is Hypericum cumulicola, each of these flowers should be a seed. From doing demography on Hypericum, we found that some of the biggest seeds can have more than 2,000, or some of the biggest plants can have more than 2,000 seeds. And then with multiple plants per square meter, and then the accumulation of seeds over time, we just expected to find more. So one reason that I was thinking about was maybe there's really high seed predation. Animals are just coming in and eating a lot of them, or maybe they degrade more quickly than we thought, and the seed bank isn't as persistent as we thought. But it would be interesting to look into that further. And then a few more questions that I had just after finishing this study were, how does time since fire affect the seed bank? Does seed bank structure shift in the dry season? What is the maximum depth from which seeds can germinate in the scrub? And how do species richness and genetic variation of the seed bank in the two habitats and with time since fire compare? Acknowledgements. Thank you, Erin, for all of your help and support during my project. Haley, Alon, Sterling, Kelly, Scott, Lexi, and Christine, thank you for being such great lab mates. Vivian and Julie helped me a lot with GIS. All the interns I've overlapped with, you've been really great people, and I've really enjoyed getting to know you. And Archbold and the Von Jordan Foundation, thank you for funding and support. Questions?
All right, thanks, Ella. That was an awesome talk. We got a whole bunch of questions. Let's see. Abe says, thanks, Ella. Nice study. Eric Mangus asks, why do you think there are fewer seeds in roadsides? Fewer seeds produced, more loss of seeds to germination or burial?
I have no idea. I mean, the questions that we listed were maybe they degrade quicker, maybe they produce more seeds. I kind of would lean towards producing more seeds because maybe it's a more stressful environment, but I could see all of those possibilities being true.
Great. Alon asks, have other studies also found that most seeds occur in the first 4 to 6 centimeters of soil? AKA, is your result supportive of current published research?
There have been a lot of studies on active seed banks, and the paper that I found that talked about the active seed bank compiled a lot of different studies. So they said that there was a range of the top 1 to 5 centimeters was found in other places.
Great. Abe has a question this time. What's known about seed longevity of scrub obligate seeding species?
Seed longevity is believed to be pretty long. Like I read somewhere that around 30 years, I think, for a lot of species.
Let's see. Tori says, great work, Ella. How or why did you choose those 2 species to study?
I chose those 2 species because they're obligate seeders and they're a type that I was interested in, but they're also pretty prevalent throughout the scrub. And it was easy to find some good roadside and interior habitats for both of them.
I'll also mention that you actually chose 3 species.
I did. One of them didn't work out so well. I also chose Orangium cuneiform, but those ones did not, I didn't find any seeds for those.
Julie asked, do you have a favorite scrub plant species?
Ooh, probably Baldwina because it looks really happy.
Cool. And it looks like we have a few more in the chat. I'll just read a few of them off. So Jack asked, do you think seed predators could explain how few seeds you found?
Yeah, definitely. I know the plant lab might be leaning towards a predator seed experiment, but I won't spoil too much there. But I would be really interested to see that answer. It's one of the reasons I could think that seeds are just disappearing.
Yeah, and for these 2 species in particular, I know someone had done a study with them showing that like 30% of Paronychia seeds were removed and like 60% of Hypericum were removed. So it's definitely a possibility. Let's see, Jessica asks, how might the flooding that occurs in the wet season influence the seed bank density or location?
Oh yeah, no, the paper that I read about the bimodal distribution was really cool because there's some seeds that are buoyant and they aren't really affected by that, but the seeds that are buoyant kind of float back towards the surface, like in where the cracks were in the wet season.
Yeah, great. And yeah, Eric mentions a few of the seed bank papers from the plant lab. So you can check those out in the chat. All right. Well, thank you, Ella. It's been great having you in the lab for the last 8 months. And it's been a excellent presentation. So
Thank
I'll
you.
hand it back to
Thank
Laura
you.
then.
Oh, yeah.
Okay, thank you again, Ella, and thanks to everyone again for attending today. Please watch our website, our newsletter, and our Facebook page for information on our upcoming seminars for this year. And we will see you soon. Goodbye.